At some point in your mid-20s, without any announcement, without any single detectable moment, your body switched directions. Everything that had been building bone density, muscle mass, lung capacity, the speed at which your cells repair damage, the precision with which your immune system identifies threats peaked and began, quietly and without stopping, to go the other way. Not because of anything you did, not because of your diet or your sleep or your stress levels. Those things matter at the margins, but the underlying process, the biological machinery that determines how fast you age, was already running before you had any say in it. For most of human history, we accepted this. We called it aging, we called it natural, we called it inevitable. Then, in the last 2 decades, something changed. Scientists stopped treating aging as a fact of life and started treating it as a disease, a specific, mechanistically understood, potentially treatable disease. And what they have found in the biology of why your body starts declining at 25 and why it cannot stop on its own has produced some of the most extraordinary and controversial science of the 21st century. Most people think of aging as a single process. Things wear out. Cells get old. The body winds down. That is not what the biology shows. In 2013, a team of researchers led by Carlos Lopez Otin at the University of Oviedo published what became one of the most cited papers in biology of the decade. Published in the journal Cell, it identified 9 distinct biological processes. They called them the hallmarks of aging, each one a separate, mechanistically distinct way that the body deteriorates over time. In 2023, they published an updated version adding 3 more hallmarks, bringing the total to 12. 12 separate biological processes, all running simultaneously, all interacting with and accelerating each other, all beginning in measurable documented ways in your mid-20s. Aging is not one thing going wrong. It is 12 things going wrong at once, and understanding even a few of them changes how you think about the body you are living in right now. In 1961, a cell biologist named Leonard Hayflick at the Wistar Institute in Philadelphia made a discovery that the scientific establishment initially refused to believe. He was culturing human cells in a laboratory, growing them in dishes, allowing them to divide and reproduce when he noticed something that contradicted the prevailing assumption. The cells did not divide indefinitely. After approximately 50 divisions, they stopped. They did not die immediately. They entered a state of permanent arrest alive, metabolically active, but no longer capable of dividing, no longer capable of replacing themselves. This became known as the Hayflick limit, and it is now understood as one of the foundational mechanisms of biological aging. Every time a cell divides, it must copy its DNA, and every time it copies its DNA, the ends of the chromosome's protective caps, called telomeres, get slightly shorter. Telomeres function like the plastic tips at the end of a shoelace. They protect the chromosome from fraying and losing critical information during copying, but with each division, the tip gets a little shorter. Eventually, it becomes too short to protect anything, and the cell receives a signal to stop dividing permanently. The discovery of this mechanism and of the enzyme telomerase that can, in certain circumstances, rebuild telomere length earned Elizabeth Blackburn, Carol Greider, and Jack Szostak the Nobel Prize in Physiology or Medicine in 2009. Your tissues are full of cells that have reached their limit, cells that stopped dividing years ago, cells that are no longer replacing themselves. The tissue around them is aging because the workforce that maintains it has quietly retired and cannot be called back. But the cells that stop dividing don't simply sit quietly. In 2008, biologist Judith Campisi at the Buck Institute for Research on Aging documented something that has since become one of the most active areas in the entire field. Senescent cells, the cells that have hit their Hayflick limit and stopped dividing, do not retire peacefully. They become toxic. They begin secreting a cocktail of inflammatory proteins, enzymes, and signaling molecules into the surrounding tissue. Campisi's team called this the senescence-associated secretory phenotype, the SASP. The SASP is, in biological terms, the equivalent of a factory worker who can no longer do their job, deciding to spend the rest of their time sabotaging the machines around them. The inflammatory signals released by senescent cells damage neighboring healthy cells. They disrupt normal tissue function. They recruit immune cells that cause further inflammation. And most disturbingly, they can push neighboring cells into senescence themselves. One zombie cell creating more zombie cells. A slow, self-propagating wave of cellular dysfunction spreading through aging tissue. In 2011, A team at the Mayo Clinic, led by Darren Baker, published a study in Nature that confirmed what this meant in living animals. When they cleared senescent cells from aging mice using a genetic technique designed to selectively destroy cells, expressing a senescence marker, the mice lived significantly longer. They developed fewer age-related diseases. Their tissues showed measurably less deterioration. The implication was direct and historic. Aging, at least in part, is being actively driven by cells that are still alive. Remove them, and the process slows. They are not a symptom of aging. They are one of its engines. In 2020, a clinical trial led by Greg Fahey at the Intervene Immune Research Group published results in Aging Cell. A small group of participants received a combination of growth hormone and 2 diabetes medications. After 1 year, their epigenetic clocks had moved not forward, but backward. On average, participants' biological ages decreased by 2.5 years. It was the first clinical evidence in humans that the epigenetic aging clock is not a one-way dial. David Sinclair is a professor of genetics at Harvard Medical School and one of the most controversial figures in modern aging research. His central argument, developed over 2 decades and published in his 2019 book Lifespan and in numerous peer-reviewed papers, is that aging is not caused by the accumulation of damage to DNA itself. It is caused by the loss of information about how to read that DNA. Here's the distinction. Your DNA is like a master copy of a book. Every cell in your body has the same copy, but the epigenome, the system of chemical marks that tells each cell which pages of that book to read, is different in every cell type. A liver cell reads different chapters than a heart cell. A neuron reads different chapters than a skin cell. As cells age and divide and encounter damage, those epigenetic marks become disrupted. The reading instructions get scrambled. Cells begin expressing genes they were never supposed to express, and silencing genes they were always supposed to keep active. The result is not just cellular dysfunction. It is a gradual loss of cellular identity. Old cells begin to lose their specialization. They become, in a measurable molecular sense, less distinctly themselves. Sinclair's hypothesis is that if you could reset the epigenetic marks, restore the original reading instructions without changing the underlying DNA, you could reverse cellular aging. His lab has demonstrated this in mice. In 2020, his team published results in Nature showing they had restored vision in old mice by resetting the epigenetic age of retinal cells using a combination of 3 genes. The cells did not just stop aging, they became younger. The mice's vision measurably improved. The experiment did not extend the mice's lives, but it demonstrated in a living mammal that a specific age tissue could be biologically rewound. In 2009, a landmark study published in Nature produced a finding that stunned the aging research community. A drug called rapamycin, originally developed as an immunosuppressant for organ transplant patients, already approved by the FDA, already in widespread clinical use, extended the lifespan of mice by up to 14% when administered late in life. The mice were already 600 days old when treatment began, the equivalent, in human terms, of starting an intervention at around 60 years of age, and it still worked. Rapamycin works by inhibiting a protein called mTOR, short for mechanistic target of rapamycin, a master regulator of cell growth, metabolism, and autophagy. Autophagy is the cellular cleaning process by which cells break down and recycle damaged components. It is one of the body's primary mechanisms for removing the molecular debris that accumulates with age. As cells age, autophagy slows down, debris accumulates, function declines. mTOR, when overactive, suppresses autophagy. Rapamycin, by inhibiting mTOR, restores it. The National Institute on Aging's Interventions Testing Program, the most rigorous independent longevity drug testing program in the world, has since confirmed the rapamycin finding across multiple independent laboratories using genetically diverse mice. It is currently the most robustly replicated longevity intervention in mammalian biology. Human trials are underway, clinical data is being gathered, and a growing number of longevity physicians are already prescribing it off-label, a fact that has generated enormous controversy inside and outside the research community. Back to the senescent cells, the zombie cells that accumulate in aging tissue and drive inflammation. If clearing them from mice extends healthy lifespan, the obvious question is whether we can do it in humans. The answer, as of 2024, is we are trying. A class of drugs called senolytics compounds specifically designed to identify and destroy senescent cells while leaving healthy cells intact has moved from animal studies into human clinical trials. The most studied combination is dasatinib and quercetin, dasatinib being a leukemia drug, quercetin being a plant-derived flavonoid. Together, they selectively trigger apoptosis, programmed cell death in senescent cells. A 2019 pilot study at the Mayo Clinic, published in eBiomedicine, administered this combination to patients with idiopathic pulmonary fibrosis, a fatal lung disease strongly linked to cellular senescence. After just 3 weeks of intermittent dosing, patients showed measurable improvements in physical function, walking speed, and 6-minute walk distance. It was a small study, preliminary, incomplete, but it was the first evidence in living human beings that selectively clearing senescent cells produces measurable biological benefit. Larger trials are now running across multiple institutions. Alzheimer's disease, diabetic kidney disease, COVID-19 long-term effects, all conditions where cellular senescence is believed to play a driving role, all being tested against the same basic intervention: remove the zombie cells and see what the body does next. Here is where the science leaves us today. Your body began its decline at 25, not because of bad luck or poor design, but because of specific, named, mechanistically understood biological processes that evolution never had a reason to fix. 20 years ago, the question was, why do we age? The answer to that is now largely known. The question scientists are asking today is different, and the fact that they are asking it seriously, in the pages of Nature and Cell and the New England Journal of Medicine, is something that no generation before this one could have said. The question they are asking is, does it have to? And for the first time in the 4 billion year history of life on this planet, no one is certain the answer is yes. Please subscribe to this channel if you are enjoying these bewildering facts.